Welcome to Careers in Discovery, where you'll meet scientists who've forged outstanding careers in biotech and hear about what they've learned along the way. This podcast is brought to you by Singular, building brilliant biotechs. This week on Careers in Discovery, I'm absolutely delighted to be joined by Paul Thompson of Mission Therapeutics. Paul, welcome to the show.
Yep, thanks, Tom. It's nice to be here. Looking forward to the chat we're going to have.
Absolutely, great to see you. So I always start by talking a little bit about what you're up to now, Paul. So please tell us a little bit about Mission, and for those who might not know, about Dubs and the work that you're doing there.
Right. Yeah. So I'm at Mission. I've been here for about 7 years. The company has existed for over 11 years. It was founded back in 2011 by a very esteemed cancer biologist, Steve Jackson, who'd previously founded a company called Kudos that was responsible for development of the PARP inhibitor olaparib.
Right.
And he had a great idea about this novel class of enzymes called DUBs, deubiquitinating enzymes, and their potential role in DNA damage response. And he thought, why not let's try and set up a new company and based around this biology, see if we can get some chemistry and then target these enzymes and see if we can't find some disease opportunities for these, primarily in cancer at the outset of the company. So that was quite a long time ago. That was maybe 4 or 5 years before I joined Mission. And during that time, I think Mission was really trying to establish the chemistry platform, the proprietary chemistry platform that enables us to target these enzymes, and then also try and develop some of the cancer biology stories at that time. I think it's fair to say that DUBs are not an easy class of enzymes to target with chemistry. And because they're novel and the understanding of these enzymes in human biology is limited, I think Mission has been doing a couple of tough things in parallel, developing new chemistry and working out new biology at the same time.
Right. Okay.
So to take a step back, these enzymes, what do? They're responsible for removing ubiquitin chains off of proteins. And it's thought that that's important in controlling the fate of the protein, whether that be through ubiquitin being a degradation signal, or whether it being a signal for different activity of the protein or localization of the protein. And so by these enzymes taking ubiquitin off, they can modulate quite a few key functions of the protein and obviously outcomes from pathways.
Right.
And the idea behind trying to inhibit them is that you might be able to build up ubiquitin on proteins that you want to clear from the system, or you might be able to shift activity of proteins based on the outcome that's desired. And so that's the general concept of trying to inhibit DUBs, is to change protein homeostasis in the right way for a certain disease setting.
Yes.
So it's a very interesting area of biology because it's so new. The chemistry is new as well. So as I said, a couple of challenges in parallel, but it's a lot of fun.
Yes, I can imagine. And if it was easy, someone would have done it by now, right?
That's true.
Yeah. And so your role there, Paul, is you're the Chief Scientific Officer, of course. Tell us a bit about, I suppose, where you spend your time and what your focus tends to be on.
Right. Yeah. So I'm primarily responsible from a line perspective from a lot of the biology work that goes on at Mission. So the target validation team here at Mission is in my group, and the translational science team at Mission is in my group, and bioinformatics. So those 3 key areas are really my line responsibility. I probably have a couple of direct reports from those, but I also have a couple of manager direct reports that also have direct reports into them. So I've actually got quite an interesting blend of managing managers and managing lab scientists.
Right. Okay.
So that's actually quite interesting and probably keeps me a bit more up to date with the challenges that continue to be experienced in lab research. So that's one side is the department, and then another part of it is leading the programs. So I've had responsibility for the early discovery programs whilst we were uh, building them from target validation stage. I've
Hmm.
since handed over those program leaderships to other folks in the department. But I still maintain a very strong link to the main programs that Mission's been pushing forward over the last 5-6 years, and those are programs targeting a mitochondrial enzyme called USP30, which is thought to be involved in mitochondrial quality control.
Right.
So I lead a program that has a CNS penetrant USP30 inhibitor, and we're moving that forward for Parkinson's disease. And then I also, of course, having spent so much time thinking about USP30 biology, I'm also part of the team that's a little further forward, which has a peripheral compound against USP30 that's in the clinic and is moving forward in kidney disease and other diseases. So there's quite a lot from the program side, quite a lot from the department management side, and then of course as being part of the exec leadership team at Mission, and being a small company, we're always out looking for fundraising
Yeah,
opportunities.
of
So
course.
there's a lot of time spent together with the rest of the leadership team, getting our pitch right, um, conducting presentations with prospective investors, um, discussing our situation with our current investors. So plenty of that. So yeah, I'd say three main buckets
Mm.
of work, and all quite different. And as I said, all very, very enjoyable.
Yes, and plenty to keep you busy from the sounds of
Yeah,
it.
it has been busy. I'd say Mission working in a small company. This is my first small company, like
Right.
biotech. Um, but it definitely has been a lot of hard work. But of course, the payoffs could be huge from a
Hmm.
medicine development perspective, and I think that's uh, that's why we do this kind of job, I think.
Yeah, absolutely. And I want to come back as we go through this conversation to that idea of moving into a smaller company, because I think,
Sure.
that's something that people have perceptions around, and some of those perceptions are accurate and some are not. And it'd be useful to understand your experience around that. But
Yeah.
I also wanted to ask you, Paul, and we'll talk about this, I'm sure, as we talk about your career. You've come to the chief scientific officer role from a kind of circuitous route almost. You've trained as a biologist, obviously, but then you've spent a lot of your career prior to this in clinical research. And I believe, if my research is right, you actually joined Mission as a medical director, so different focus. Um, how how do you think that has informed the approach that you take in that role? Because a lot of the chief scientific officers that we speak to have really spent their entire career focused on this early research and haven't
Mhm.
necessarily ventured into that later stage development. So just interested to understand your perspective on that.
Yeah, circuitous is a good word. Uh, yeah, that's right. So I did spend some time in industry at the bench at GSK in
Hmm.
the first job that I had. But after that, yeah, I really wanted to see application of the science in the clinic.
Yes.
So I think it was probably definitely a driver that I had that made me go down this route, actually, rather than something that organically happened as such.
Mm.
Um, and through that, I have been able to see, yeah, the application of science in early clinical development plans. I've also had the opportunity to look right to the end of medicine development. So having responsibility for phase 2 programs
Right.
that are global, and sort of thinking much more about the end.
Hmm.
So what are we doing this for? What's this medicine going to deliver to patients? How's it going to address unmet needs? What about payers? What about physicians? What's their perspective? And I think that is a very healthy thing to understand as you then go back and try and build programs from the early stage, because it
Yes.
can give you a different perspective. Um, there may be upsides to that. They may be downsides to that, actually, that you think you're too far, you're taking one step too far too early,
Okay.
if you see what I mean,
Yeah.
thinking about the end too early. But that's something that I'm conscious of. So
Mm.
whenever I'm working with the discovery teams and the early target validation work and target ID work that we're doing, really have to still keep open, not get too bogged down about thinking about the end. Of course, there
Yes.
has to be a destination in sight, but not to the extent that it restricts or let's say um, makes a call too early on a target story without having given it the opportunity to show exactly what it can do. So I think it is quite. So I've definitely found it very helpful for me, obviously, and so now in the CSO position, I'm able to I'm able to influence the early programs in the way that my experience tells me should be done, but I'm also able to think very proactively in the programs that are at that transition to clinic about the narrative and actually
Right.
what's the purpose of this whole program, which
Yes.
ultimately is benefit to patients and can society pay, right? So and I think this is so as I said, been very helpful for me, but it's not something that was organic. I kind of wanted to
Mm.
experience the end-to-end process.
Yes. Okay. Interesting. Well, let's talk a little bit about that then, about sort of the origins of all of that. So, I guess we'll go right back to the beginning first of all, Paul. For you, why science? Why drug discovery, drug development? Why this career for you?
Okay, yeah. So I suppose that that I mean, I've been interested in science. I was interested in a lot of things,
Mm.
but certainly growing up, but certainly having the biochemistry degree from Oxford and then doing a PhD in London. And the PhD that I did in London was a MRCK studentship. So had the opportunity to spend some time at GSK for 4 or 5 months
Right.
in the final year of that studentship. And that was a good experience, I think, to be exposed to a supervisor that had a perspective on medicine development
Hmm.
and learn about the ways that science can be prosecuted in an industry setting compared to an academic setting. I think that was probably more influential on my choice at that point than I thought of at the time,
Right.
actually. The PhD that I did was interesting. It was in leukocyte transmigration, great lab in UCL and Ridley's team. Um, but I think I did find the experimental progress, quite slow.
Okay.
That could have been me. I don't know. But but I found that it would be nice for things to move at a slightly faster pace generally.
Mm.
And then having had that exposure to industry through the case studentship was really helpful. So I had the opportunity actually to join GSK straight after the PhD studentship finished,
Yes.
and I took that opportunity, and I think it was, yeah, as I said, thinking continuing on that theme of wanting to understand and be part of the drug development process
Hmm.
was definitely a factor at that time. And
Yeah.
then from then on, yeah, as we sort of mentioned in the previous segment, bit of time at the bench and then transitioning to clinical research.
Yeah. Okay. So you'd had a taste of industry in, inverted commas, I guess, as part of your PhD, and I guess that's something that not everyone has. So that had given you an insight into what happens there. Um, what are your sort of early memories of sort of arriving at GSK full time and being at the bench there and working on the programs there? What are the things that I guess surprised you or that were memorable, or what sticks out?
Yeah, so that's a good question. The unit that I joined it was actually a lab that was part of an experimental medicine unit within GSK, so up here at Cambridge, and the lab had maybe a bit of freedom let's say, to try and problem solve. So thinking about, let's say, new mechanisms for existing medicines, or trying to understand liabilities of existing medicines in the lab, and generally as a potential path to assist the main programs in GSK. So there was still a lot of, I'd say, still a lot of freedom for investigational science at that stage. But I think that I was also more aware that this needed to bring some value quickly.
Right.
Because otherwise the potential path of that investigation would be stopped.
Yes. Okay.
So that was definitely the first experience was quite a good one. Still some freedom to do some interesting science, at least in that context. And you have to remember, this is quite a long time ago. All industries doing interesting science at the moment, but back in those days, it was maybe we had a bit more freedom than other departments in GSK, but certainly the idea that there had to be fairly clear outcomes within a reasonable time.
Yes.
Otherwise the progress would be paused and shifted on to something else, and that's a very healthy mindset, I think, generally, and a good one to experience early in any career, actually. I suppose not necessarily just drug discovery. But I think that was very, very useful. I think also we at that point we did have quite a few folks that joined GSK that were pretty experienced academic investigators, so professors and professors of medicine, professors of immunology from really high caliber institutions in the UK, and that provided an opportunity to understand their perspectives and how that blended into the pharma environment. And being in this group in Cambridge, an experimental medicine unit, we're exposed to both that. So I think, again, a bit fortunate, really, to get that experience with those folks that joined.
Yes.
But again, it definitely enabled me to do something probably, I think, quite exploratory for us at the time, and I can talk in more detail about that. But that as you wish. But certainly having that experimental medicine approach within industry, I think was a really great experience for me.
Yeah, and it's interesting. It'd be great to hear more about it. Yeah, because I think there was a period of time, I guess, probably a couple of years ago, where a lot of companies were starting to look at repurposing, right? And what else you could get out of compounds, or maybe neglected compounds that were just on the shelf somewhere, and that sort of stuff. It seems to have sort of fallen out of favor a touch, I think, perhaps because of pressing infectious disease issues, but which in itself was, you could argue, was a repurposing of something, right? But yeah, tell us a bit about how you were approaching it then.
Yeah, I guess the so the description of the early lab stuff that I was talking about, we were looking for new mechanisms and things, and I suppose that could, as you said, that could be considered as repurposing. I think, just thinking about the word repurposing, we were actually in the experimental medicine unit, we would try to, let's say, repurpose methods and apply them to the drug development setting. So I think in line with the experience that I previously referred to, but stopped short of going into, we had a really bright professor who went to a conference, saw a wonderful method called stable isotope labeling and its application in neurological disorders, and brought the knowledge back. And we thought, okay, maybe we can apply this to some of our programs in GSK, particularly for Alzheimer's. And that sort of passion and sort of commitment to the idea from someone very bright coupled with someone like me who was quite, let's say, young but keen, but could appreciate the potential impact of
Right.
this for clinical development, allowed us to form quite a nice team to try and mobilize parts of the GSK machine to support this experimental medicine approach. And for me, that was a really good learning experience. So it enabled me quite early to try and be very clear about why
Mm.
we want to engage and use resource from safety assessment, from CMC or chemistry development, for a program that is essentially building a biomarker for a clinical development plan, and that was quite exploratory.
Yes.
So really needing to get the message across what value it could bring, and try and sort of make that vision very clear to individuals that potentially weren't thinking about this all the day, every day, which is again sort of a very common experience now,
Yeah.
in what we do. So that, I think, was such a valuable experience, to understand how to mobilize resources, and then also the power of the team. So what you could do if you got more than just yourself trying to do something.
Right.
And that
Yes.
was the first experience that I had of really doing that across cross-functional areas.
Hmm.
And it took a while, took a couple of years for us to actually um, enact the approach in a clinical setting. But for me, it was a fabulous experience. And again, something probably quite unique, brought about through being at an experimental medicine unit within an industry
Mm.
setting, and having the input from bright academics,
Yeah.
that could assist. So I was probably
Yeah.
quite opportunistic in taking it, right? But I certainly wanted to do that.
Hmm.
And as I said, I think it's a good experience for me.
Yeah, and I suppose it gives you a really strong foundational understanding then of how these programs work, right? If you're looking for opportunities to implement this method, you've developed a deep understanding of the method, and then you're having to get your head around, well, what are you actually trying to do at each stage of this process, and look for opportunities to apply it. And I think that's probably a really nice um, base to then go, okay, well, these are the areas that I think I can have an impact on, or I'd like to be part of, or I'd like to learn more about. It broadens your perspective, I suppose.
I think that's such an important thing. Is to have your eyes open, yeah,
Mm.
to other things, other than just what's in front of you. And yeah, that kind of whole project management, let's say, or project building experience definitely opened my eyes
Yes.
early to safety assessment and chemistry development, and etc. But I think as I progressed through career and started leading clinical teams, and then leading project teams, you do obviously have to interact with more functions than just
Yeah.
safety and pharmacology and um, chemical development. And it does give you a much broader appreciation of how many things need to happen to get something done, and how important every part is in making sure that a project can deliver. And I guess, yeah, I think that was a valuable learning for me. And I think that said, I was probably very open to that.
Hmm.
And it depends on your perspective, right? I think um, if I'd been more focused on the lab research and really
Right.
enjoyed doing lab research and bench research, maybe I wouldn't have transitioned to that kind of broader experience early.
Yes.
But that was for me something that I was quite happy to do.
Yeah, it makes sense. And as you say, that led you into working in clinical research. And I'm interested in kind of that transition for you, because I think there is a perspective or an assumption that clinical research is, and people can't see this because we're not using the video, but I'm going to use inverted commas here, is somehow less scientific
Oh yeah.
than drug discovery. And I think there's also a perspective that it is just a process and it's project management rather than anything else. And of course, there are elements there, right? But yeah, what what caught your eye about clinical research beyond, obviously there's patient impact there, or you're closer to the patient, certainly. What caught your eye? Tell us about that transition, and I guess what should people bear in mind about if they think a move into clinical research might be for them? What are the
Yeah.
things that they should maybe think about?
So I think yeah, the perspective that you put in the inverted commas, I think there is a certain amount of procedure following in clinical research.
Mm-hmm.
Of course, there has to be because it's
Yeah.
regulated. But what for me was interesting, I think going back to that example that I just described, we were essentially trying to measure pharmacodynamic effects of a compound. And pharmacodynamic effects are an output of the biological mechanism that you're
Right.
pushing. So actually, for me, it wasn't such a big step to think, okay, well, we do this in animals or in cells.
Mm.
Actually, the most important place to do this is in a patient.
Yes.
Because if you can demonstrate that in a patient, then you can understand whether your drug's moving the pathway and so on. And so my route into clinical research was solely thinking about, okay, the mechanism that we think this drug works through in cells and in preclinical systems. I want to demonstrate that in patients because that's a very key early de-risking step
Mm-hmm.
to move on to the longer term, more expensive outcome studies in phase 2B, phase 3. So for me, that was the driver. And so the group at the experimental medicine unit that I was in, not only were we looking at fluid phase biomarkers, but there was an imaging group. So looking at CNS imaging through PET radiochemistry
Yes.
and MR-based approaches, and having research being more focused on neurological disorders for me, again, this was perfect. So I could see the application of these methodologies in early clinical development, speaking to these bright folks that could tell us how to use them and what the upsides and limitations of those technologies were. And so, yeah, that was it. For me, it was application of what you're doing preclinically into the clinic as a
Yes.
de-risking step for the longer term outcomes. So whether that be through biomarker team or a translational medicine team, technology team, that was the way that I found the route into clinical research. And then of course, after you've used biomarkers or imaging assessments in the first clinical study or two, then you start thinking, oh, actually, it's quite interesting to do the broader piece. And so that's how you develop. That's how I developed into leading clinical teams rather than just being a biomarker
Yes.
representative on a clinical team, if you see what
Yes.
I mean. So it kind of just grew from there. But I hope that, I hope that example is useful because I think we in drug discovery we're all trying to um, build narratives, disease narratives with evidence. And first, we typically do them in preclinical settings, but
Yeah.
ultimately, it's important, obviously, to be able to do that in the clinical setting.
Yeah, certainly. And I suppose this probably only happens a few times in people's careers if they're lucky. But I imagine there is a point in a clinical trial where you get a readout or you get some data and you realize, oh, this actually works. Which must be great, right? And that must be a fantastic moment.
Right. So it's just another experiment
Mm.
in a way. Okay, it's bigger
Yeah.
and more expensive and well regulated and includes patients, and so the ethical requirements are much greater and
Yes.
the safety components around it. But ultimately, it's the next experiment, of course, it's also very gratifying to see an experiment in vitro work actually, and
Yeah. True.
and an
Yeah.
animal disease model work. There's still nothing that I like more than seeing a positive outcome from any
Mm-hmm.
experiment. But you're right, if it's in the clinic and it's taken 2 years and however many patients' time and commitment, and you see something that comes out and it's worth it, yeah, it
Yeah.
is quite gratifying. Um, And I think that's one of the great parts of working in drug development and in industry is you can be part of making a medicine, and everyone at every stage of that process has done something valuable for that medicine development. That's something that's gratifying, I think, for anyone that's been part of that.
Yes, I can imagine. And then, so fast forwarding a few years, obviously you spent some time with GSK, you were there for a number of years, and then moved into a company that, not a small company, but a slightly smaller company, but I imagine very different culturally. Tell us a bit about that transition for you.
Yeah, that was interesting transition actually. I felt that I'd spent a long time at GSK, looking forward to new challenges. I'd spent a lot of time working in neurological disorders, and working as a clinical leader or project leader, and thought it might be useful for me to get even closer to the day-to-day in clinical research, actually.
Yes.
So the smaller company, Ono Pharma, they're obviously a large, one of the oldest Japanese companies. I think it might be the oldest. I hope anyone that's from that company listening forgives me if I got that wrong. But they had headquarters over in Japan, and then an office in the UK that essentially took responsibility for clinical development in the UK and EU. And I, through that office, we were essentially operating as their clinical development arm. And of course, in a large company like GSK, or a large pharma company, you've got many people in those departments. So you're only generally interacting as a project leader with the clinical leader or the clinical ops leader. You don't really get to see the day-to-day. And I think that was what I enjoyed seeing up in London at the Ono team there, and also broadening experience outside neurology in clinical development. So we worked on quite a few different programs across different indications. So that for me was a good next step, broaden therapeutic area experience and more day-to-day clinical development understanding. So yeah, a genuinely good experience. Culturally, yeah, slightly different, but in the end, I think the same principles apply in that we're trying to make sure that everything we do drives value, and the value can be supported by evidence in a reasonable period of time, and the medicine's likely to do something for a patient that isn't necessarily available to them today. So yeah, the same, very similar, really.
No, absolutely. And then, so if we move on to 2016, so I imagine you must have been sat there thinking, I'm making way too much money and I've got far too much spare time. I'm going to join a biotech company. I'm joking, of course. But this was your step into a small company, as you said. This was your first sort of early stage company. So 7 years ago, where was Mission as a business? What was the situation?
Yeah, so 7 years ago, Mission had just raised a Series C funding round, and there was some optimism that Mission could move their lead programs sort of quickly into clinical development, and of course and personally, when I looked at the opportunity, I was very excited about working on USP30 as the mitochondrial dub, but also very excited about the potential application of dub inhibition to targeting proteins involved in neurodegeneration. So going back to the very beginning, we talked about these dubs taking ubiquitin off proteins. We inhibit them. That means we accumulate ubiquitin on proteins and potentially force them for degradation. And in neurological disorders, many of them are characterized pathologically by protein accumulation. So in Alzheimer's, it's A beta and tau. In Parkinson's, alpha-synuclein. And so the idea that we could actually increase ubiquitin on these aggregates and force the cell to clear them through the lysosome, that sounded like a really exciting opportunity to me. So I was I was intrigued by both the lead program and the potential of the platform to do something very different for neuro disorders. So
Yes.
that's what got me excited about coming to Mission back in 2016. So yeah, that was where we were, Series C, thinking maybe within a few years we'd be able to get our first compounds through preclinical tox and into the clinic.
Yeah. And so what were your what how much did it meet your expectations of joining a small company, and how much were things a surprise? Tell us about that transition, because I think it is very different, right? It's a very, very different set of resources, set of expectations, operating environment. Yeah, tell us what you remember.
Yeah, I remember. Yeah, it was different. So so a lot less of the large department support, of
Hmm.
course. Maybe some increased focus on delivery within a time frame. Yeah, and I think so. So I think those were two things that were took a while to get used to.
Mm.
I think at the same time, there were a small group of committed scientists to this idea, so that was really good. I think the sort
Yes.
of team element from the small company perspective was obvious at an early stage. So I felt that was a pretty let's say welcoming scientific environment to move into.
Hmm.
I think also then as you're working with teams in the small company, you really get a good sense of connection actually with a group.
Right.
So yeah, if you get it right, of course, you can still have the same high performing team characteristics as you can at any other
Hmm.
company. So I felt the team side of the small company was really good, and not a transition actually.
Right.
Probably slightly, maybe slightly improved. I don't know. Um, but I think that yeah, less sort of larger department resource support was different. That said, given what I'd worked in previously and the journey that my career had been on through into clinical development, seeing quite a lot, and also having touched on the lab work before, I was fairly comfortable that I had the wherewithal going into a small company that I wouldn't necessarily be stuck because there wasn't sort of so many people being able to
Yeah.
support. I was fairly convinced that I knew enough at that time to take the jump into small company where I would be responsible for projects at that time. So I kind of felt I could,
Yeah.
I understood enough.
Do you think, and this may be a difficult question to answer, I'll explain why I'm asking first of all. So I spent the first 10 years of my career at a really large recruitment companies, not the same scale as GSK, but it was maybe 3,000 people in different countries and whatever. So
Yeah.
in recruitment terms, it's a big business. And I'd always had this sort of thing in the back of my mind that I was going to go and start my own business. But when I left there, I didn't feel confident in my own ability versus sort of the resources that were around me to go and do that. So I went to a different company for a couple of years before I did that. Do you think you would have been as confident if you were coming straight out of GSK into that environment without
Yeah,
having to
no.
take a step?
So I think you're right. I think the fact that I transitioned almost through a smaller
Mm,
company, a smaller entity in the UK, at Ono, I think definitely assisted me because yeah, I was looking to come in and take a program into the clinic. Um, my experience at Ono gave me the day-to-day clinical know-how,
yeah.
operations know-how that I could come in and do that job, which I hadn't seen at GSK. So yeah, I think the transition, kind of the stepwise transition from very large to more
Mm,
medium size, with us being at a local department, then into biotech was quite useful for me. But uh, maybe other people might be more straightforward to just jump straight in.
yeah, I think sometimes people, especially in big companies, I think people underestimate how much they know. because everybody else around you knows, right? So you've all
Yeah.
had the same training and you've had similar experiences and that kind of stuff. I think it's, especially if you've not been in a smaller environment, it's easy to underestimate how much that knowledge doesn't exist always in every company.
Yeah.
So yeah, I think, bearing that in mind is helpful, because I know it makes people a little bit nervous when we speak to them about leaving a very large company for a very small company, because
Mm.
they've been told it's extremely risky and
Yeah,
and
I'm not sure. I mean, I haven't been a large company for a long time, but things do change in large companies
yeah,
as well. And I think one of the upsides of being in a smaller company is you have more visibility to decision making. That doesn't necessarily mean you can influence it straight away, or you can understand it obviously straight away. But you do have more visibility to the workings of a
yes,
business, which I think can put things into perspective um, a little bit better than sometimes you might in a larger company that makes a big strategic decision, and
yeah,
changes your work focus as a result. So I think I, I personally I really enjoyed that part as well, being close to sort of understanding the day to day from a business operations
yeah,
perspective. Um, yeah, so I think there are a lot of upsides working in a smaller company. That said, I think my training came, and I did the inverted. My training came in the big company, right?
yeah,
So as I was at GSK, I was through that journey fortunate enough to have not just the bright academics that joined the companies, but some very, very experienced clinical development folks. So early clinical physicians that I learned so much from.
mm,
Pharmacology team department leaders that I learned an awful lot from. Um, and I think, if I was thinking about, I would probably do the training at the larger company
yeah,
than at the smaller company. So transition that way around, I think, um, would be my perspective.
mm,
Um, that said, again, in a small company in the right environment, we've been fortunate to have um, very experienced folks leading groups. So again, you can tap into their knowledge very
yes,
easily as a young scientist coming in. So actually, you can get a faster look at a lot more in a smaller company, but maybe you're not necessarily able to see so many experiences across those. You just get the one program or the
yeah.
two program focus that a small company might. So yeah, pros and cons to both. I think that both are well, can be very good fun and great learning opportunities. But I like to think that understanding the nuts and bolts at large companies probably makes sense.
I think it's a really interesting point, because I think there has been, over the last probably maybe 10 years, certainly 5 years, a bit of a sort of, not just in biotech, across every industry, a bit of a sort of glamorization of startup businesses and small businesses and this sort of stuff, right? And it can be very exciting. And as you say, there are massive upsides if you get into the right company and it grows, or, you get access to people that you wouldn't get access to normally, and what have you. But I know people who are CEOs of small biotech companies that are maybe 50 or 60 people, in which you would go and talk to the CEO, who have run departments of 10,000 people, that you would
Hmm.
just never see that person, right? So there is that. I think, though, it doesn't necessarily logically follow that big companies are worse to work for. I think I think it's different, and some people wouldn't suit the big company environment. And similarly, some people wouldn't suit the small company environment, right? But I think it's, as you say, you can still surround yourself with really amazing people in either one, and it's finding that that's key.
Yeah, you can, and I think that in a large, it really depends what you like doing. So
Yeah.
I mean, the most important thing is to enjoy what you're doing, and that normally means you're doing something well, I think. So if it's you're in the lab and you're enjoying your research, and you're enjoying generating data from preclinical systems or in vitro systems, that's great. Keep enjoying it. Have a basis of expertise actually within the industry that
Mm,
allows you to build. So as soon as you're sort of, yeah, senior scientists, principal scientists, getting onto larger teams, let's say early program teams. Delivering well for that team can open up opportunities. And I think if you're delivering well, it normally means you're enjoying what you're doing.
Yes.
And then from there, if you really like to be still doing lab research, that's great. If you want to see different things, you created the opportunity to do that. And I think you can do that very well at large companies. Yeah. So I agree. I think I mean I maybe I was fortunate at GSK. They had some training programs that were very useful. And I'm sure it's the same at other large pharma companies. Sort of the potential to understand, let's say, product labeling strategies, which you wouldn't necessarily see at a small company, but you could get on a course and understand what the finer points of early clinical pharmacology and how that builds into a drug label. Those opportunities were fabulous. Again, for me, I'm probably fortunate. So I think there's a lot to be said for understanding the basis of drug development in large companies. That said, you can also do it the other way around.
Yeah, absolutely. And I think if you, particularly if you're a practical learner, right? If you learn by doing things, then sometimes the small company might be the right environment because you're going to get to do more stuff within that focus. But yeah, I think understanding your own preferences is important for this, and people underestimate that, I think.
Yeah.
So Paul, we're going back to mission and where you are today. So there's lots going on. Tell us a little bit, I appreciate some of it will be confidential, but tell us a little bit about what's next, what the next 12 months holds, where you're heading with it all.
Yeah, so the next 12 months are pretty exciting for mission. So we have the lead asset that's the lead USP30 inhibitor that's been through phase 1 healthy volunteer studies. So now it's getting set up for phase 2 patient studies. And so again, we get back to that part of development that we talked about earlier of seeing effects in patients, biomarkers, whether it maybe some imaging, but then outcomes. That's the next step for that program. So yeah, for me, that's a very exciting point. And then for the CNS program, that's just a little bit behind. We hopefully take that one to the clinic for first in human studies starting early next year. So again, a very exciting time for us for that CNS program. And having worked on the USP30 program here at Mission for 6 years, seeing this progressing on is really, really good.
Yes.
But then, of course, also some of the earlier programs that we're coming through from the discovery team, we go through in vivo POC studies the second half of this year for a couple of programs. And if there are positive outcomes there, we can signal those are transitioning to lead up programs, and then it gets pretty exciting. So a lot of hard work that's gone on in Mission the last 12 years is coming to a point where it's very, very exciting. So yeah, lots of good stuff to come, hopefully.
Excellent. So a really pivotal time.
Yes.
And we wish you the best of luck with it, Paul. Thank you so much for joining us today.
Yeah, no problem. Thanks, Tom.
Thanks very much for listening. Careers in Discovery is sponsored by Singular, helping you to build a brilliant biotech company. Biotech leaders spend far too much time, money, and energy on hiring and people issues. Head to www.singular-biotech.com to learn how you can recruit and engage your team more effectively so you can focus on developing medicines, treating patients, and saving lives.